On behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor, Almac, I'd like to welcome you to Discovery, Development, and Commercialization of Multiplex Diagnostics and Companion Diagnostics. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. At this time, I'd like to introduce our presenters for today's webinar. Michael Sloan is Vice President of Business Development for Almac Diagnostics, where he has served since 2005. He has full responsibility for the overall management of the company's business development activities and driving the growth of the company. Michael has a Bachelor of Science in Commerce and a Master's of Science in Corporate Leadership and was educated in the University of Birmingham, Napier University, Scotland, and the Ivey Business School, Canada. He has over 20 years in senior business development and management roles in the biotech and medical device sector. He's worked in both startup and multinational companies. His previous experience, including Managing Director for BSN Medical UK, as Smith & Nephew, and Beiersdorf JV and Executive Director for Amtech Medical Limited. Our second presenter is Dr. Richard Kennedy, who is the Medical Director of Almac Diagnostics and is responsible for the application of the company's technology into medical practice. Dr. Kennedy became the Maclay Professor of Experimental Medicine at the Queen's University of Belfast in July of 2011 and leads an academic research group which focuses on novel approaches to biomarker discovery and delivery. Richard is a practicing consultant in medical oncology at Belfast City Hospital and is involved in running first-in-man clinical trials. Prior to coming to Almac, Richard worked as an instructor in oncology at Harvard Medical School, where he specialized in translational cancer research, and he continues to work as a medical oncologist at Belfast City Hospital in the UK. He's currently a member of Dundalk Institute of Technology and has previously served on the CR UK Biomarker Panel and Enterprise Ireland Life Sciences Technology Panel. With that, I'll turn the presenter ball over to Michael. Welcome.
Okay, thank you very much, Elizabeth, and thank you everybody who's joined us this afternoon or this morning, wherever you're dialing from. So the topic of the webinar today is the discovery, development, and commercialization of multiplex diagnostics and companion diagnostics. The overall purpose of this webinar is we at Almac believe that in order to be successful, or for personalized medicine to be successful, it's going to require a further shift to developing more complex assays in order to measure and more complex biology. This is already happening, and during this webinar what we want to do is to provide you our overview of that shift, but more importantly examples of what we've done and how this can be done and how these more multiplex assays can be discovered, developed, and actually commercialized as well. The agenda for today, I'm going to give you a very high-level overview of Almac Diagnostics and talk about why we believe multiplex assays are the way forward. And then Richard Kennedy is going to give you some detailed case studies on our DNA damage repair deficiency assay and our angiogenesis diagnostic assay and how we've discovered these assays and developed them also. And then finally, I will wrap up with some of our thoughts around the delivery models and commercialization models for these types of companion diagnostics following development. Okay, so firstly, very high-level overview of our business. Almac Diagnostics is part of a group of companies called the Almac Group, which provide integrated solutions for drug and diagnostic development from research to commercialization, made up of 5 separate businesses, all providing different services to pharma and biotech companies. We're headquartered in Northern Ireland, where all the divisions are represented. We also have a US headquarters in Pennsylvania, headquarters in Singapore, and an office in Tokyo. 8 facilities worldwide. The company is financially stable, privately owned, over 30 years experience serving over 600 clients with approximately 3,500 staff. These are our locations. Okay, so Almac Diagnostics, so what do we do? Well, we are very much focused on the discovery, development, and commercialization of multiplex diagnostics and companion diagnostic assays. Our business model falls into 2 strands. We have what we call our contract biomarker services, where we're working with pharmaceutical and biotech companies as their diagnostic discovery, development, and delivery partner, and then we have our own internal biomarker research. In terms of our services, they fall into these broad categories from assay development. We run trial enrichment studies through our CLIA lab. We do biomarker trial management. We offer bioinformatics consultancy. We're running retrospective trial collection where we collect samples not collected during clinical trials to enable biomarker profiling post the completion of the trial. We have laboratory services, and then ultimately we're partnering with pharmaceutical companies as to develop companion diagnostics. All of the platforms that we have in-house are all essentially selected because of their ability both to measure and to commercialize more complex assays. I'll not go through this, but they fall into DNA, RNA, and then we're supporting services such as bioinformatics, sample sourcing, pathology, IHC, etc. And in terms of our internal research, we have a number of different assays at different stages of development and commercialization. Some of these which Richard will present and talk you through today to give you the background of how they've been discovered. Our most advanced is a stage 2 colon prognostic assay. This assay was published in the JCO in 2011. It's an assay to predict the likely recurrence of stage 2 colon cancer post-surgery. Since that publication, we have actually licensed this assay to Precision Therapeutics for distribution and sales in the US. We have done a further validation study in a CALGB clinical trial, which was presented at ASCO GI, again showing a very robust hazard ratio of 2.1. And now this assay is now commercially available via Precision Therapeutics. It's called now the GFX colon assay. The second assay is our DNA damage repair deficiency assay, which Richard will talk about in some detail today. This particular assay, the details were published in the JNCI in January of this year. The clinical utility is to predict response to standard chemotherapy, anthracycline-based chemotherapy in breast cancer. And at this stage, we've licensed this assay to Genomic Health for exclusive global distribution in breast cancer, and we're working with them to perform further validation studies now. We also have a very interesting new assay that's just validated in ovarian cancer. It's our Angiogenesis assay, which again Richard will talk about in some detail. And we have behind this then a prostate cancer assay, which we're in the process of validating now. So that's just a very high-level overview of our business and our business model. So in terms of today, why do we believe that multiplex assays are important to the success of personalized medicine? Well, basically, if you look at the field today and the companion diagnostics available today, this slide really outlines the successes to date. So there really are 19 in total, of which 10 of these assays are actually HER2. So there's only really actually 6 biomarkers that have been commercialized as companion diagnostics, which are listed there. They're all oncology, they're all DNA, protein, fairly simple tests. And I think it's fair to say that but despite these successes, in terms of personalized medicine companion diagnostics, there's been limited success really to date. What we have learned is that the target is not always the test. A good example of this is the EGFR inhibitors, for example, cetuximab and panitumumab, where the EGFR receptor is not a strong biomarker of response to these drugs. And in fact, it was the KRAS mutational status that's much more important for predicting response. So this just tells us you can't rely on the target as a biomarker of response for your compound. The other thing we've learned is that the biology we're looking for is complex, and therefore the diagnostic we need to develop will need to be complex. The underlying biology of a disease and response-resistant mechanism to chemotherapeutic is complex. A great example of this is the angiogenesis inhibitors that are targeting the VEGF pathway. Neither the VEGF receptor or the pathway predict response to these anti-angiogenics. And I think you'll see when we present the information on the assay that we've developed, which is a complex assay, that the reason for that is that it does require us to go in and look for the more complex biology and be prepared to discover and develop on platforms that can do this to enable us to find these biomarkers that we've long strived to find. So our conclusions really from these things are, firstly, that it's had limited impact, but I think certainly the opportunity is great for personalized medicine. Companion diagnostic successes have been simple and assays and kit-based. The easy biology has already been identified, and that's what's been presented today. For the industry to evolve, the field must move towards these more complex assays, angiogenesis being an example, as I've already quoted. Multiplex assays for complex biology, in our opinion, will be the future for diagnostic and companion diagnostic success. So that, I hope, really sets the scene, I guess, for the message and the information we want to share today. I'm now going to pass to Richard, who will now give you some fairly detailed case studies as to how we've done this in some of the assays we've developed.
Okay, thank you, Michael. So I'm going to cover two of the products that are in our pipeline today, the DNA damage response deficiency assay and the angiogenesis assay. Talk a bit about the background, how we came to develop these assays, and sort of principles we'd applied to finding these biomarkers. So first of all, looking at the DNA damage response deficiency assay, or DDRD for short, I'm going to talk a little bit about chemotherapy for breast cancer, just to orientate you, and then a bit about the development of the assay. I'm a breast cancer medical oncologist. When I treat patients, I typically use a combination of DNA damaging agents, usually a combination of something like cyclophosphamide and doxorubicin being an anthracycline. The problem is, in the clinic, I really don't know who's likely to benefit from the treatment and who is not, and because of this, we probably overtreat. The single agent response rates are actually quite poor for these agents. They're around about 20%, so there's a lot of people not benefiting from them. And our problem as clinicians is we really don't know who benefits. So when we're treating patients, we tend to apply some kind of algorithm for risk stratification. So in the UK and Europe, traditionally we've used algorithms such as Adjuvant Online or the PREDICT. More recently, we've had access to Oncotype DX, which obviously has been available in the US for considerably longer. But each of these assays categorize patients as low risk, intermediate risk, and high risk. Low risk patients, the risks from chemotherapy outweigh the benefits, so we usually do not recommend chemotherapy. Intermediate risk, there may be a marginal benefit for chemotherapy to prevent disease recurrence. So in this case, we discuss chemotherapy with patients, and usually there's about a 50% uptake. In patients who are high risk, these are typically patients who have lymph node involvement or high-grade disease. We would typically offer chemotherapy as there's a significant risk of the disease coming back. So one thing we do know, we've known over a number of years, is that patients who have an abnormality in the Fanconi anemia BRCA pathway, so they may have mutations in BRCA1 or BRCA2, or loss of different parts of this pathway, seem to respond better to chemotherapy. And indeed, this is probably the underlying mechanism for chemotherapy response in breast cancer, so it could be a good biomarker to guide response to DNA damaging agents. The problem we have is that there's no reliable method for detecting loss of this pathway in the clinic. We could certainly sequence for BRCA1 and BRCA2, but that's only a small part of a very complex pathway. So I'm not going to go through this in depth, but this is just to give you an idea just how complex this molecular pathway is. And this is from a review which I did about 7 years ago with Alan DeAndrea. Really, the purpose of this pathway is in normal cells, you have to deal with endogenous DNA damage all the time. This pathway forms a complex of a number of the Fanconi anemia proteins, which then ubiquitinate the D2 protein, which forms a second complex which is involved in repairing the DNA damage. And this is particularly important in repairing the damage that occurs following cross-linking agents, such as cyclophosphamide or anthracyclines. And following this, the damage is repaired and replication is allowed to proceed again in the cell. Loss of any part of this pathway will make a cell very sensitive to chemotherapy, and about 30% of solid tumours lose this pathway, probably as part of the genomic instability that is required for cancer development. Now, the problem is that when we come to develop a biomarker for this pathway, it can be difficult. I've highlighted here some of the components of the pathway that have been reported to be lost in solid tumours, and you can see it's quite diverse. And these are not only lost through mutations, they can also be lost through epigenetic mechanisms such as methylation, or can be lost through alterations in the ubiquitination pathway. So there's multiple ways that these things are lost. So it makes developing a biomarker very, very difficult. Our hypothesis was, although there's lots of ways you could lose this pathway, the cells still have to be able to survive the endogenous damage which can occur through metabolism. So there may be a common response pathway in Fanconi anemia BRCA deficient breast cancer, and that could act as the biomarker. So rather than worrying about all the ways you get to loss of repair, maybe there's a common response pathway to loss of this repair. Also, we know that tumours with known BRCA1 and BRCA2 mutations, at least some, should display this pathway. And also, and this is something we've probably done a little differently to the field, is we went back to the disease Fanconi anemia itself. So patients with Fanconi anemia, which is a rare homozygous mutation for one of the Fanconi anemia genes, patients that have this mutation are very, very prone to DNA damaging agent. And we thought, well, if they have this and they carry this mutation, then their cells should also be demonstrating the survival pathways that allow them to survive loss of the pathway and this could help us identify tumours which have also lost this pathway. So we took a dual approach. We acquired a data set with collaboration with the Mayo Clinic of over 150 breast cancer tumours that had been enriched for BRCA1 and BRCA2 mutations, and we performed unsupervised clustering in this data set. At the same time, we got a data set of over 20 patients with Fanconi anemia. These are bone marrow aspirates from these patients with matched controls, and we identified the molecular processes that were activated in the patients with Fanconi anemia. And then we combined the data sets and looked for the molecular subgroup in breast cancer which had demonstrated the same biology as seen in patients with Fanconi anemia. As far as molecular subtyping goes, we standardized each gene to the median pro-set expression value. We performed hierarchical clustering using Pearson correlation distance and Ward's linkage. And the number of subclusters were determined using a statistical approach, which is the gap statistics, so it's a robust method for identifying molecular subtypes. So this is looking at ER-negative breast cancer, and if you look at the highlighted molecular subgroup in the middle, but just to orientate you, the x-axis are tumour samples, the y-axis is gene expression, red is upregulation, green is downregulation. And you can see in the middle group of tumours, there's upregulation of a distinct biology, which I've highlighted. And importantly, this group was significantly enriched for BRCA mutation. The second, when we look at ER-positive, the exact same gene cluster comes up as activated. And when we look at the groups where this biology is activated, again, we find there's enrichment for BRCA mutation. So this group is of potential interest. So the next thing is we found this group is anything to do with loss of Fanconi anemia BRCA pathway. Now, as I mentioned earlier, we'd also looked at the molecular processes that were activated in patients with Fanconi anemia, and we found that these were identical genes and identical processes to the ones that represent these clusters where we're seeing BRCA enrichment. So it seems these molecular groups are just demonstrating the same biology as you would see in patients with Fanconi anemia, and this was highly significant. We thought, well, perhaps this group, an ER-negative and ER-positive, represents loss of the Fanconi anemia BRCA pathway, and therefore we developed a classifier that could identify this molecular subgroup prospectively. And I think there's an important point here. A molecular subgroup itself is not a diagnostic, it is not a biomarker. Sometimes you'll see this in the literature where people will have presented heat maps like these with molecular subgroups and say they've discovered a biomarker. This could not be used in the clinic because it relies on a large data set to be analysed to find this subgroup. You still need to have a biomarker or assay that could be applied to a sample on a one-by-one patient basis as they come into the clinic to categorise them into the subgroup. So we developed a classifier. It was a 44 gene signature that would put people in this molecular subgroup, which we thought represented loss of the Fanconi anemia BRCA pathway. We called this the DDRD assay for daily damage response deficient. So far we've identified a molecular subgroup in ER-negative and ER-positive breast cancer, which was defined by the same processes. And I haven't got into this today, but it's mostly a type of immune biology, which is quite interesting. It's immune biology which seems to be activated by loss of DNA repair. The question was, could this subgroup represent a biomarker for breast cancer therapy? So the next thing was to independently validate the biomarker. So we took a dual approach. First thing was to look in the neoadjuvant setting. So these are patients who have received 6 cycles of 5-fluorouracil, epirubicin, and cyclophosphamide chemotherapy prior to breast cancer surgery. Typically, this is used in patients who have fairly large tumours, and we wanted to decrease the size to allow surgery. And this gives us a very good readout for the predictive value of our assay because we have a response metric. The other data set is an adjuvant data set. So these are patients who have had surgery and then have had the chemotherapy to prevent disease recurrence. So from this, we can assess the prognostic relevance of the assay in the context of chemotherapy. So first of all, looking at the neoadjuvant setting, this was applied to 203 patients who received neoadjuvant treatment. The endpoint was pathologic complete response, which is quite a stringent endpoint. And from this, we found that patients who were DDRD positive on the multivariate analysis had an odds ratio of complete pathologic response of 4. So they're about 4 times more likely to get a complete response to chemotherapy. And this was very significant. Importantly, the assay had never seen this data set before, so this was a true independent validation. And also, if we look at the multivariate analysis, we can see that the DDRD assay was independent from other factors such as estrogen receptor, grade, and stage. Then looking at the adjuvant setting, so to remind you, these are patients who have had surgery and then followed by chemotherapy to prevent disease recurrence. In this case, we ran this through our diagnostics labs and what we call a prospective retrospective independent validation. So I know that seems quite a strange term, but what this means is it was run as if you were running the test in the clinic. The sample was sent to us, we gave a positive or negative result, and then it was unblinded at the end to us. to see had we predicted how patients had done following chemotherapy. These were patients where we had at least 5-year follow-up, so the outcomes were known. And the assay was applied to a single 10-micron FFP, so it's formalin-fixed paraffin-embedded section that had been macrodissected, so this was clinically relevant material. And what we found was, if we look at this graph, so this is relapse-free survival, the top bar, or the top graph, the blue, represents a DDRD positive population, the red represents the DDRD negative, and you can see patients who were DDRD positive had significantly less recurrence following DNA-damaging chemotherapy, with a hazard ratio of 0.37, so they're about 3 times less likely to develop disease recurrence in 5 years if they receive chemotherapy. Importantly, we also have to make sure that the assay is not just telling us something we already know, so we ran a multivariate analysis, and again the assay was independent from other factors such as ER, age, stage, grade, HER2 status and nodal status. So it was a true independent prognostic factor in the context of chemotherapy. There are a number of prognostic assays in breast cancer, so it was important to make sure that this was actually not just another prognostic assay that was telling us something about chemotherapy. So we analysed 664 patients who had breast cancer who had not received DNA-damaging chemotherapy, and in this case you can see that the assay was not prognostic. So the assay is only prognostic in patients who have received DNA-damaging chemotherapy, which would fit with the underlying biology, which is the Fanconi anemia BRCA pathway, which we know responds to chemotherapeutic damage. So how do we think this could impact in clinical practice? So if we go back to the diagram I gave earlier, we tend to categorize patients into low risk, intermediate risk, and high risk. Now we think when we apply the assay for patients who have the option of chemotherapy, if they're DDRD negative, there's probably no point in going forward with chemotherapy. So rather than having a discussion in these patients who are believed to have a sort of a moderate benefit from treatment, there's a population here who probably don't get benefit from these agents, whereas those who are DDRD positive should probably have the treatment. So we've taken that subjectivity out of that decision. For low risk, we could argue that patients who are DDRD positive, it may be worth having a discussion because now you've actually pulled out a population who may actually respond to treatment. So there may be a proportional benefit for these patients, whereas those who are negative, again, probably should not have treatment. In the high-risk population, who should receive some form of treatment, the DDRD positive, we believe should still receive DNA-damaging chemotherapy. Those who are DDRD negative, these may be the population where we should be adding in additional agents which work outside DNA damage, such as taxanes. And there's actually some data we have to support that at the moment, and we're currently running a clinical study to further validate that. So just to give you sort of what's happened to this assay, it was out-licensed to Genomic Health for use in breast cancer this year. We're currently working with Genomic Health on further validation studies to get this ready for the clinic within hopefully the next year. But this is an assay which we really hope will impact patient care over the next few years. The second assay I'd like to talk about is our angiogenesis assay. So this came out of a program which was started with Charlie Gurley, Professor of Medical Oncology from the University of Edinburgh a few years ago. And the idea here was that in high-grade serous ovarian cancer, treatment hasn't changed for over 20 years. Patients usually have surgery followed by paclitaxel and carboplatin chemotherapy. And unfortunately, the majority will relapse within 5 years, and about 70 to 80% of patients, unfortunately, will die within 5 years. So there's a lot needs to be done for this disease. So the aim here was to identify the molecular subtypes of high-grade serous ovarian cancer using unsupervised hierarchical clustering of gene expression data, then to determine the biological pathways and processes that characterize these molecular subtypes, and then try and predict the therapies that might be best for specific molecular subtypes in ovarian cancer. To discover the subtype, we ran molecular subtyping, so we profiled 287 formalin-fixed paraffin-embedded specimens. These are patients who had been treated in the University of Edinburgh. They had standard debulking surgery for their ovarian cancer, followed by carboplatin and paclitaxel-based chemotherapy. Now, importantly, all these samples were reviewed by 2 expert pathologists. This is important because historically, samples have been misclassified as high-grade serous, so modern diagnostics, histopathological diagnosis has changed, so it's important for doing this kind of work that they are reclassified. And I think that's one of the strengths of this particular piece of work, that this pathological review was carried out. 2 10-micron sections were macrodissected and sent to Almac for analysis. And it was analysed on the Almac diagnostic ovarian cancer disease-specific array. This is a version of the Affymetrix platform that has been optimized for formula-fixed paraffin embedded tissue. It also has additional transcriptional information on it that we developed in-house from deep sequencing of ovarian cancer. So it has a very good coverage of the transcriptome for ovarian cancer. The other important thing is that this was done in a very controlled manner. We made sure that there were no batch reagent effects, there were no user effects, anything that could affect our ability to pick up molecular subtypes. And unfortunately, a number of the molecular subtyping approaches that have been carried out in cancer over the years have been confounded by technical issues such as reagents or site or user or technology. So using this then, we performed a molecular subtype analysis of these high-grade serous ovarian cancers. So these are the molecular subtypes that were identified, and this was presented at the American Society of Clinical Oncology about 3 weeks ago by Charlie Gurley in the gynae plenary session. And what you can see here is the 3 major molecular subtypes, and the major biologies that seem to define these are immune signaling and angiogenesis signaling. So if we take the first group here, labeled C1, we can see that the genes for angiogenesis are turned on, the immune turned off. In the C2, the second group, we can see that the genes for angiogenesis are off and immune is turned on. And in the third group, both immune and angiogenesis are turned on. For the purposes of this presentation, the most interesting group is probably C2, where we see that angiogenesis is inactive. So we've discovered molecular subtypes. The question is, did they actually tell us anything? So because this was a well-annotated data set from Edinburgh, we were able to look at the progression-free and overall survival for these patients following conventional treatment. And what we found was, if we first of all look at the progression-free survival graph, the green line represents C2, the angiogenesis inactive group. The red and the blue lines represent the angiogenesis active groups. And as you can see, the angiogenesis inactive group for progression-free survival has a considerably better outcome than the angiogenesis active group, with a hazard ratio of 0.74. Interestingly, the 2 groups with angiogenesis active tend to track together, so it seems to us the angiogenesis was the major discriminating biology when it came to phenotype for the molecular groups. If we look at overall survival, again you can see that the angiogenesis inactive group has a better overall survival than the 2 groups with angiogenesis, with a hazard ratio of about 0.66. This was significant. So overall, the angiogenesis inactive group seems to be prognostically better than the angiogenesis active groups. I suppose that kind of makes sense in that angiogenesis, you probably expect to have a worse outcome where the tumour is growing in some blood vessels. So at this point, we identified 3 molecular subtypes defined by activation or repression of genes related to angiogenic processes. And patients in the subtype with inactivated angiogenic gene expression have better survival following standard chemotherapy. So the next question, or what we thought at this point, is we potentially had identified a prognostic assay in ovarian cancer, which represented probably about 40% of the disease. So this was interesting. Similar process to what I showed with DDRD. This is just molecular subtyping. We still had to develop a classifier that could identify these patients prospectively in the clinic. So we identified a 63 transcript signature using a partially square approach that could separate this group from the 2 groups which had angiogenesis. So for the purposes of going forward, the angiogenesis inactive is the C2 group in this diagram. Angiogenesis active is the C1 and C3 groups grouped together, as they seem to behave much the same. So the next question is, we have this prognostic assay, but do we see this when we look at another data set? Can we validate this phenotype? So the first thing we did was an in silico validation. This is data published by Tothill in Clinical Cancer Research about 6 years ago. They had 152 high-grade serous ovarian cancer samples. Again, patients who had surgery followed by conventional treatment with paclitaxel and carboplatin. These were profiled on the Affymetrix +2 platform. This was useful to us as our platform is compatible with this, so we were able to run our assay on this data set. And also the progression-free survival and overall survival data was also available for this data set. So this is a data set our signature had not seen before. When we applied it, again we can see that the angiogenesis inactive group, as demonstrated by green here, had a better progression-free survival and overall survival when compared to the angiogenesis active groups. And again, if we look at the hazard ratios, they're comparable to the previous data sets. Hazard ratio of 0.65 for progression-free survival, significant. And overall survival hazard ratio of 0.34, again, highly significant. And importantly, this is a multivariate analysis, so we factored in other prognostic factors, such as age, stage, grade, and debulking status. So this is a true independent prognostic assay. And actually, in my time in ovarian cancer, I think this is probably one of the most powerful independent prognostic assays that I've seen to date. So the obvious question is, this is all defined around angiogenesis. So does this go to tell us anything about anti-angiogenic agents in ovarian cancer? And this has been an area of some controversy. Avastin or bevacizumab, the generic name for the drug, is available for ovarian cancer in the EU and Europe. It has been licensed by the European Medical Agency. However, the FDA in the US has not licensed it for ovarian use, although it is used off-label by the majority of clinicians in ovarian cancer. The reason that it was not licensed by the FDA was it did not demonstrate an overall survival advantage in clinical trials, only a progression-free survival advantage. So it is an area of some controversy. So our hypothesis was that patients within the angiogenesis inactive subtype maybe would not benefit from Avastin, whereas patients in the angiogenic group may see the benefit, and we may be able to use this to categorize patients for this treatment. And just to explain to you, this treatment is relatively expensive, so it would be useful to be able to identify patients who truly benefited. One of the studies which demonstrated the benefit for, or the progression-free survival benefit for Avastin in ovarian cancer was the ICON-7 study. This is a European and Canadian study which looked at carboplatin paclitaxel plus or minus bevacizumab. And so we were able to access this through the Medical Research Council, who collected a number of the samples for the purposes of translational research. So we performed a formal biomarker validation using this set. We had a pre-specified protocol. We sent this to MRC. MRC would send us a sample to our diagnostic lab. We would then do a call, which is negative or positive, send the results back to MRC, and the MRC statisticians then broke the code and did the statistical analysis. So this was a proper biomarker validation. We were in no way able to influence the results. So first of all, if we look at the control arm of ICON-7, we can find, again, we're seeing the same pattern we've seen in the other data set. So this is the third validation now that the angiogenesis inactive group has a better prognosis than angiogenesis active. The angiogenesis inactive group in this progression-free and overall survival graphs is green, whereas the angiogenesis active is blue. And you can see for progression-free survival, hazard ratio is 0.52 and significant. For overall survival, hazard ratio is 0.53 and significant. Again, this is multivariate analysis, so this is additional to the other information that we usually collect. And I think this is important because it demonstrates that there's nothing particularly unusual about the ICON-7 data set. We're seeing a very similar prognostic outcome for the angiogenesis inactive group in the control arm of this study. This next graph is probably the one which has probably got a bit of a reaction at ASCO. Now we've taken the angiogenesis inactive patient group as identified by the assay and looked at the effect of adding Avastin. So first of all, we look at progression-free survival. The blue line represents conventional treatment in the angiogenesis inactive group. This is progression-free survival. The red line represents progression-free survival after you add bevacizumab to this group. And what we're actually seeing is a detriment from adding Avastin in this molecular subgroup. We're seeing that the hazard ratio is 1.73. The median progression-free survival falls from 36 months down to 18 in this molecular group when you add in Avastin. This was surprising to us because initially we'd expected there would be no benefit from Avastin, but what we're actually seeing here is a detriment. If we look at overall survival, again we're seeing the same pattern. Patients who get conventional treatment are the blue line on this graph. Patients who receive the addition of Avastin is the red line, and you can see that there's a detriment to overall survival with the addition of Avastin in this angiogenesis inactive group with a hazard ratio of 2, again highly significant. And this is multivariate analysis again, so this is factored in other clinical factors such as debulking, age, grade, stage. So the next thing to look at the other side of the coin, these are the patients who are in the angiogenesis active group. And here we can see that the addition of Avastin is showing a benefit. The progression-free survival increases from 12.3 months up to 17.4. So we're seeing the opposite effect in this molecular group. So if there is angiogenesis, there seems to be a benefit from bevacizumab. The other interesting thing is the shape of this curve. If you look, you can see that the red line represents the patients who received Avastin. But in ICON 7, the Avastin was stopped at 12 months. And you can see what's happened. After 12 months, the survival or progression-free survival curve falls markedly. So it seems that the patients were doing well for as long as they were taking Avastin. When the Avastin was stopped, as was part of this protocol, then the survival fell very quickly. So I think one of the lessons from this is probably in this group, they do benefit from Avastin, but the Avastin should probably continue. It should not be stopped at 12 months. And this kind of subgroup analysis is useful for doing this kind of analysis from a clinical trial and working out what has happened. So just to summarize, so this angiogenesis assay has been independently validated in ICON 7 as a predicted outcome following treatment with Avastin. And the angiogenesis inactive patients are adversely affected by the addition of Avastin to standard of care, whereas those who are active are showing, while there's a trend towards improved progression-free survival, it didn't quite hit significance because we don't have the entire ICON 7 study yet. It's something we're working on at the moment. We also plan to validate this finding in the other major study that, in which Avastin was used in ovarian cancer, the GOG 170 study, and we're in the process of accessing that study at the moment. Our aim would be, if we validated this in 2 independent studies, then is to be able to deliver this for predictive purposes for Avastin in ovarian cancer. The other interesting thing is because this is a molecular subtype defined by a specific biology, we're able to find that the same subtype exists in other diseases. We've found that this angiogenesis active and inactive biology is in colon cancer, breast and lung cancer. We've gained access to large clinical studies in each of these areas where Avastin has been given with them, or chemotherapy has been given with and without Avastin. We're currently in the process of validating it in these settings as well. It's one of the strengths of molecular subtyping is we tend to identify assays which are transferable across diseases. And this just leads me then to hand back over to Michael to talk a bit about the delivery and commercialization.
Thanks very much, Richard. So hopefully those case studies represent some very good examples of how understanding the underlying complex biology can enable you to find new biomarkers that can really change clinical practice. And we are actually working with multiple other programs as well with our pharmaceutical partners using the same approaches to develop companion diagnostics for the compounds and the agents that they are developing as well. But as Richard said, the other thing then that needs to be considered is the delivery and commercialization routes. So if you are going to develop a multiplex companion diagnostic, then considering the platforms that may be utilized and the methods that are employed to run those assays, you do need to step back and now consider what's the most appropriate model then to actually commercialize those assays. Really, there are 3 commercialization options. One is the LDT PMA model, and this is the lab-developed service-based model, but PMA, FDA-approved assay. So an FDA-approved assay being delivered from a service lab as a companion diagnostic. The second option is the more traditional route, which is where the assay's converted and manufactured into an IVT and distributed as a kit. Then finally, actually, we have 2 hybrid models that we employ where perhaps you initiate the development of the assay as an LDT and then migrate it to a kit at the appropriate time. In order to decide or to make the decision on what's the most appropriate model, there are a few factors you need to take into consideration. Then the main factors are firstly timelines, and what we mean by that is what time is available based on the timing of the existing clinical trials. Very often the clinical trial timelines are already set out and established prior to incorporating a biomarker strategy, and time can be tight between, for example, phase 2 and phase 3. So it is important that that timeline is understood and that your strategy for your biomarker is designed to fit that, and that it does allow time to actually prepare and lock down and validate that assay before it's utilized in the next phase of the study. The budget's another consideration. The decisions that need to be made are considerations such as, do you wish to invest in a full-blown manufactured kit prior to knowing the outcome and the results of your phase 3 trial, which could fail, and obviously there's a more significant investment in a kit strategy versus an LDT strategy up front. So considerations around budgets are important. The technology platform, again, a very key consideration. If this multiplex assay is developed on a platform that maybe isn't as readily available in every clinic in the world, then there needs to be a consideration as to how that's made available. And again, perhaps they can lend themselves more to a service-based model under those circumstances. And then final very key point is obviously forecast and the scale-up of that forecast. How many tests are going to be required, and obviously the scale of that forecast is going to affect the model. The LDT model will obviously more suitable for a slightly lower forecast, but scale up is important too, because it is possible that an assay could be launched as an LDT with a view to migrating it to a kit as the volumes increase. So these are the things to consider. The next couple of slides are just really high level to set the scene and to help people start thinking of these models. But this is some of the models that we utilize in Almac. This model demonstrates the LDT PMA approach, where the assay is eventually commercialized as an FDA-approved service-based test. And the key things to note here are the period between the assay lock at the end of phase 2. So the assay is developed during the phase 2 clinical trial, and then you require 12 months to do the verification studies to prepare that assay for utilization in a phase 3 clinical validation, and then further regulatory submission, after which it's then ready for an LDT PMA commercialization. The other thing, so the timeline for preparing that assay for your phase 3 is shorter. The cost of developing and regulating that assay is also less than the kit approach, but you do need to bear in mind the technology platform and the forecast when considering this option. The other, I guess, option, or the other extreme to that, is the kit approach. And here you'll see the big differences here are that the time between the assay lock and at the end of phase 2 and the start of the phase 3 is actually 24 months. So in order to take this approach, because you need to have your kits manufactured and ready for the phase 3, it does require an extra 24 months. So this needs to be incorporated in the strategy up front, and it's something very much to bear in mind. And very often we will find with our partners that it's this period that is challenging, and that's where some of the more hybrid models fit in. Under these circumstances as well, it is a more costly option, because you're running your phase 3 clinical reference labs, and there's more upfront on the manufacturing side as well. But these are all, again, considerations. In order to accommodate some of these challenges, there are hybrid options. This first hybrid option represents a model where the assay is actually launched as an LDT PMA and then subsequently migrated to a kit post-launch phase. Where this is valuable is that it shortens the timeline between phase 2 and phase 3, and enables you to get the assay regulated and validated and to market. And it also reduces the cost, and that you haven't invested in the full-blown kit until you know the outcome of phase 3. Following the end of the phase 3 trial, then you would utilize the original clinical validation samples to form an assay migration and prepare the kit for manufacturing. You would have to undergo a regulatory amendment and then launch the kit as well. But this does facilitate and support some of the challenges around timing. And if the scale-up of the assay requirements is such that this fits, then the launch as the LDT migrating to kit can work very well. And then the final option is where you actually it's kind of like a catch-up strategy, where you actually initiate the clinical validation using an LDT approach, but this time you would validate the assay to CLIA and run the CLIA assay in the phase 3 clinical trial. And then in parallel to that, you would perform the assay migration and manufacturing to kit. Thereby you're getting your phase 3 underway within the 12-month window, and then you're catching up during the phase 3 as you migrate your LDT CLIA assay to a kit. And then subsequently you regulate the actual kit and launch the kit. It's the more expensive of all the options, because you're both preparing an LDT CLIA assay as well as the investment in the migration to the kit. However, it does enable you to catch up that timeline prior to the launch of the assay and the drug, and enables you to launch that assay as a kit, and that is a desired strategy. So those are really presented just to, at a high level, enable people to start thinking about some of the different options for commercialization and the models going forward. So that is our I guess, the final slide. Just, I guess, from my perspective, on behalf of Richard and I'd like to thank everyone for joining us. Just in conclusion, I guess what we're saying is we thoroughly believe, as I hope has come across today, that multiplex diagnostics will be the future of personalized medicine. We've developed our own pipeline of multiplex products, which I hope demonstrate the ability to do this and the ability to really make a difference using these approaches. We have the tools and expertise in-house now to develop and deliver these multiplex diagnostics and do so both for our own products and for our pharmaceutical partners. And we hope that we'll be considered a partner of choice for multiplex companion diagnostics. So that is the end of our presentation today. I guess we hand back to Elizabeth now, and we're happy to take questions for the remainder of the time. And thank you again for joining the call today.
Thank you very much, Michael and Richard. We do have several questions that have come in. For those of you who are with us, you can still chat your questions in, and we'll do our best to get to them. Why have you chosen a microarray platform versus alternatives such as DNA-seq and so forth?
Yeah, so I'll speak to that, Richard. One of the issues is that using DNA mutation or sequencing, well, where it's good for maybe where the drug and the mutation go together. So, for instance, if we have a mutation in something like EGFR, that might in some ways tell us how some of the targeted therapies may work in that pathway. Where we have complex molecular subgroups, a single mutation is usually not enough to identify it. And in fact, using sequencing will be very difficult to identify the molecular subtypes we've talked about today. One of the nice things about using transcriptional data is it is the final readout from a number of abnormalities within a tumour. So, for instance, if you had a mutation in something like PI3K, a mutation in PTEN, a mutation in AKT, the final transcriptional readout will be much the same. So if you use a transcriptional-based approach, it will pick up all these mutations around a similar biology. If we take actually a good example too would be the DDRD assay we presented. That's picking up, we know that's picking up mutations in BRCA1, BRCA2, probably a number of associated proteins in the Fanconi anemia pathway. It's picking up epigenetic silencing of some of the genes in the pathway. So it's picking up a lot of complex biology and grouping it together from a common transcriptional program. So certainly for molecular subtyping, I think you do need to take a transcriptional approach.
The other thing I'd add to that is NextGen as a platform, certainly from FFP and for RNAs, it's just not quite ready for commercialization yet, whereas the Affymetrix microarray platform is clear. They have the diagnostic instrument, and it is a good, robust platform for commercializing assays from.
All right, a question from Wendell. Which party typically initiates development of the companion diagnostic? Is it the diagnostic development company, the doctor, the pharmaceutical company?
No, well, in our experience, it's typically the pharmaceutical company. So we are now, it's finding that in the field now, all the pharmaceutical companies will have biomarker teams with the strategy of discovering biomarkers alongside all of their new compounds. So very typically, when we engage with a pharmaceutical company, they will come to us with biomarkers that they've discovered, and they will drive the strategy. Our role in that will be to take those discovered biomarkers and do the assay development and then validation, etc., and take them from them and I guess convert them to a product. So it's driven by the pharma companies. Where it slightly differs is in some of our own assays that we've discovered and developed. Essentially, some of these fundamental biologies, such as the angiogenesis assay and the DDRD assay, are potentially companion diagnostics in their own right, and therefore the actual discovery and presentation of these biologies, I guess, could drive interest from pharma to work with us to utilize our assays as companion diagnostics. But I think it's fair to say that to date, it's coming from inside the pharmaceutical industry that there's a desire now to incorporate these strategies and then they approach companies like us to support them in the diagnostic development and delivery side.
Thank you. Sabata asks, are these considered laboratory-developed tests?
Yes, they are, yes. The assays we've presented today are all laboratory-developed tests, LDTs, but we're in the traditional commercial model for the LDTs is to launch them as CLIA assays through a CLIA lab, not FDA approved. But what we're presenting here are models where we're actually what we're calling the LDT PMA route, where these are FDA approved lab developed tests, and we have a number of programs already going through our business where we're developing these as companion diagnostics for pharma.
Okay, and a follow-up question. The time frame for completion of the validation of the DDRD assay, I believe I saw the information on one of your slides, but we have a question wanting to know about that time frame.
So, do you want me to speak that? Yeah, no, the time frame is estimated sort of 18 to 24 months, but typically what will be required here at a very high level is a further validation study, and then there will be all of the pre-commercialization activities, impact studies, the dossier for reimbursement, etc. So these things can take some time, and it can be dependent also on your time to obtain samples for further validation, etc. So a rough estimate for actual commercial availability when a test will be on the market and reimbursed is hopefully in the region of 18 to 24 months.
Okay. What is Almac's strategy for protein markers?
Well, our core business is very much still around genomic RNA-based biomarkers. However, and that is in line with the whole multiplex strategy and our whole model here. However, we have invested in IHC in the business. We do have a number of assays running in our CLIA lab and trial enrichment studies that are incorporating an IHC assay being run. The reason for that is it's more feedback from our partners, where more than often there's a combination of assays run to make an enrichment decision. So there could be a PCR assay run first, followed by the IHC assay in parallel in order to make the enrollment decision. So that led to us bringing IHC in. So it's there very much more as a support platform to be used in incorporation. Our main focus is on the more complex multiplex platforms.
Okay, it looks like we're coming up on the hour now, so we do have several more questions, but I will make sure that they're forwarded on to our presenters today. I'd like to thank Michael and Richard and also Almac for their willingness to present today and to sponsor today's event. I'd like to thank all of you for taking an hour out of your day to come and meet with us and have an opportunity to hear about this new technology and hopefully find some ways to solve problems in the work that you're doing. So with that, I'd like to say thank you again to everyone. I hope you have a great day and a wonderful upcoming weekend. Thank you, and bye-bye.